see, which would be
The last one.
yeah. Very good.
Is that it?
Yeah, great, thank you. It was said a few years ago that if Maxwell, the famous physicist of the 17th century, had walked into a physics laboratory today, he would not recognize it at all. But if Louis Pasteur had walked into a microbiology laboratory, he'd be quite at home. That has changed because microbiology now is recognized that we as human beings are walking populations of microorganisms in our gut, on our skin, in nasal passages. We carry microorganisms, and it's been found that there's a relationship between these microorganisms and a variety of chronic and infectious diseases. So the genomic revolution has reached microbiology, and I don't think in a few years Louis Pasteur would be very comfortable in the new laboratories of modern microbiology. The fascinating aspect of it is that genomics has allowed us to understand that the bacteria in our gut produce chemicals that affect our thinking, affect our well-being, not just in terms of health and disease, but also in terms of depression, euphoria, etc. So microbiology has reached a stage where we really need new analytical tools. So over the last 5 years, a team with whom I've been working have developed a new approach to be able to identify microorganisms instantly, accurately, and with great precision that can be applied and utilized to solve infectious disease problems, as well as understanding the microbiome of all of us as human beings. So with the approach of being able to identify using genomics within minutes all of the bacteria that are in the gut and be able to predict and understand the infectious agents that we may be carrying, this allows us to identify pathogens where current tests simply don't work. That is, the tests are not able to identify particularly strains or species. So the approach of being able to determine what kinds of bacteria are present on skin or in the gut, how much of each is present, and to which antibiotics they are resistant or sensitive, doing this instantaneously or nearly instantaneously is clearly important. So this approach, in comparison, as you see on the left, where PCR techniques, that is, targeting certain genes in a given bacterium to use that as an indicator and an identifier, instead, by genomic sequencing, where we can determine all of the base pairs in the bacterial, virus, or fungal chromosomes, we are able then to determine the organisms in a way that we use the entire genome instead of just a few markers as we do in contemporary microbiology, that is using petri dishes and test tubes. This allows us then to be able to identify very precisely to the species or strain. Why is this important? Well, it turns out that E. coli, Escherichia coli, fancy name for the common bacteria in our gut, is a friendly bacterium. It produces vitamins. It is part of the natural digestive system. However, it can pick up traits. These are genes that can be transferred from one strain to another. And these traits can be quite pathogenic, so that the reports that you read about foodborne illnesses from hamburgers or from spinach, where an E. coli is implicated, is because it's picked up certain genes for invasion, for producing toxins, etc. So identifying species and strain is really important. And the current techniques are not able to do that. They can identify that E. coli is present, and then with tedious techniques over a week or so, be able to determine whether it's pathogenic or non-pathogenic. But by being able to take the entire genome sequence of the bacterium and then being able to identify by a mathematical formula just exactly what it is and to what it is sensitive or resistant is really important. This gives us a quantitative result as opposed to, well, I think it's this, and we'll try this antibiotic or another antibiotic. And with this approach, we can say this bacterium is resistant to X antibiotic and sensitive to Y antibiotic. So it's actionable, and that is really, really critical. How do we do this? The microorganisms from a sample, it can be a clinical sample, it can be a food sample, it can be a water sample for assuring safe drinking water. The DNA is extracted, and then there are many techniques now, and this is now automated. And of course, it can be sequenced. Sequencing machines now, a variety of them are available, but the approach that we've taken is mathematical and it's agnostic. Doesn't matter which sequencing machine you use. The raw sequence reads are obtained. These are then matched to a very highly curated gene library with about 15,000 microorganisms for which purified, well-known, characterized sequences are available. So the matching is done, automated, and it's probabilistic because microorganisms have a propensity of exchanging genes rather promiscuously. In fact, a single bacterium can exchange, in the case of the cholera bacterium, up to 80% of its genes can be shifted back and forth. So this means that you've got a moving target, and you've got to assess the best estimate, and it's a mathematical Bayesian approach that gives you the identified bacteria. And then matching against a library of resistance genes or pathogenicity genes for which we know their actions, then we're able to determine whether or not these particular bacteria that we've identified in this sample and identified how much they represent as part of that DNA extracted sample. And then to determine not only can we identify them down to strain, but identify what genes are present, what resistant factors they carry, what pathogenicity factors they carry. This allows us then to carry out identification of the pathogens and also to determine all of the bacteria, viruses, and fungi that comprise the flora of our gut, of our skin, of pet animals, or food, water, for any application. This is really a critical development. An example, studies of cholera patients in India and Bangladesh, a field in which I've worked for the last 30 years. By being able to analyze the gut flora, stool samples, where patients come into the hospital and we can't isolate the cholera bacterium, but in fact we know they have a very serious enteric infection that resembles cholera. With this technique, we are able, not only in the cases of where the routine clinical laboratory says, well, cholera is present, we can identify that, but where the lab says, we don't know what's there because we can't isolate the bacterium, with this technique, we can determine what pathogens are present, and as it turns out, it's not a single bacterium that's involved or a single virus. It turns out that we have to rethink infectious disease because in the case of the cholera patients coming into the lab in Calcutta, into the hospital in Calcutta, they carry from 4 to 10 pathogens. Not only do they carry Vibrio cholerae, but they also carry Salmonella, Shigella, rotavirus, and perhaps Giardia or Cryptosporidium, fancy names for a mixture of parasites, bacteria, and viruses. So infectious diseases for which we have utilized very effectively the Koch's postulates, namely you have an infected state, you isolate the pathogen, you reintroduce it to an animal model and replicate or reproduce the infection. So you've identified the pathogen, but it turns out we have to rethink the community of bacteria that act to cause the diseases, and more to the point, for those chronic infections, chronic diseases, which are increasingly beginning to turn out to have a microbial beginning when it's very difficult to identify or to be able to determine whether microorganisms is present. With this approach, we're now able, through the DNA extraction and analyzing the DNA of the bacteria, the viruses, and the fungi, we can identify when we can't culture it in the laboratory, either because we haven't got the right media or because they have growth conditions that are not replicated in the laboratory. This changes infectious disease approaches and treatment and understanding. But it's also useful to understand in healthy human beings. With the healthy control volunteers with whom we've worked in Calcutta and in Bangladesh, we've analyzed the gut flora and then drawing down from the wonderful data that's available in the human microbiome database at NIH, that NIH has funded, and the data that have been gathered around the world into the gene bank at the NIH, drawing down those data, the healthy European gut flora we find is quite different from the healthy flora of the Indian population. And you can see in the healthy control, that diagram, the pink area are identified pathogens. And the blue area are the bacteria normally introduced through probiotics, that is, yogurt and similar type of probiotic, introducing the bacteria that are protective. You can see that in the Indian population, they carry larger numbers and distribution of what would, in a Western population, result in symptoms of an enteric infection. But we think that we now see a mechanism by which our immune system is constantly triggered to protect us by the presence of very small numbers of pathogens that we carry in our gut and that act as instigators to the immune system to produce a response, a protective response. So by this genomic approach, we're learning about ourselves, we're learning about infectious diseases, we're learning also ways that we can protect ourselves by reintroducing a balanced gut flora, and probiotics and metagenomics is becoming very strongly of public interest, and I think a new type of treatment and maintaining wellness other than having to resort to antibiotics and chemical treatments. So it's a completely new way of identifying microorganisms in infectious diseases. Another way of looking at this is that you can see in the columns lined with human microbiome characterization, HMP, compared with a healthy Indian population, you can see that in the Indian population, there are known enteric pathogens which we can identify very precisely and we can quantitate for each individual. And similarly, in the human microbiome population of Americans and Western Europeans, we too carry very small amounts of a few, much fewer, but potential pathogenic bacteria, part of the natural ecosystem of our bodies. So the next generation sequencing allows us to have diagnostically meaningful understanding of disease, infectious diseases. And with this approach of being able to take the DNA sequences of microorganisms directly and mathematically identifying, binning, characterizing, and being able to provide then to the physician an actionable, precise path of treatment will revolutionize, I think, infectious diseases in the next few years. And that we can actually do this not in a huge supercomputer, but on a laptop connected to a server. And I will predict that probably in the next 2 or 3 years, we will have physicians with handheld devices with a swab to be able to extract the DNA, sequence it, perhaps with chips on the handheld, make a rough diagnostic, or simply with a click, send it to a database and get a very precise identification, not just to species, but to strain, and also to the genes present that confer antibiotic sensitivity or resistance, or infer various potential pathogenic properties. And I would predict, combined with the wonderful work being done on the human microbiome, at the same time from that sample, with the extracted DNA that includes the human, which now we discard or put aside to look at the bacterial DNA, we will, in the future, analyze the human DNA for those potential genetic characteristics that infer sensitivity on the part of the individual, perhaps to a given treatment, so that you have the full information of the bacterium, the virus, the fungus, and the human response. This will completely revolutionize infectious disease understanding and treatment, and bring a whole new perspective to human well-being, to human understanding, as well as, I think very importantly, to understanding diversity on our planet. Another topic for a discussion, but also something to think about, because now we can characterize those microorganisms, which some have said, astronomers have talked about billions and billions of stars in the galaxy. We have trillions more of these microorganisms that comprise the biosystem of planet Earth. Thank you.